So I just kind of reformulated my education.
I have a question is how much is allergy and if it, it doesn't say
I can rip stuff out. I can rip stuff out.
it's not like allergy equation.
That's how to rip stuff out. The reality is if you remember that, so as long as the pipes in and you're all right.
See, that's so simple, but that makes complete sense to me. Because you're working, if you get that big flat, you're in trouble up there. Oh, that's really good.
He's on the board.
What about you, Elise? You joining the work from?
So, I can't believe that we're headed to May already. This year has gone so fast.
It is.
Yeah,
It is. Really? That's been a year.
Now I don't see it, but I don't know. I don't know what else.
You always hear about, you see pictures of the cancer out there in the middle of the thing, and they say only certain areas can actually thrive there. So,
On the other hand, obviously
Show up.
I think it's kind of handy.
I don't know if you saw that.
I did, yeah, late last night. So, Barry Roscoe. He's a psychiatrist. He was on the faculty field, and he works in novels. The agency that David said.
I didn't have time to make it public, but UW.
Right, you don't have to go and check your passport in each country you go to.
Okay, well, that's not a private agency.
Well, I mean, he's on the faculty field, he kind of works in addition over there.
Well, no, he primarily works over there, and he happens to have a faculty.
Because he couldn't go to you.
Right.
All right,
So she should just write to him directly.
And her school is set up.
Right. Exactly. And she should just write to him directly and tell him what she's looking for, and what he can do. He's very nice.
She's studying fine allergist.
Yeah, absolutely.
Okay, good.
He's very accessible, really nice guy.
Oh boy!
Oh, just for Dr. Kennedy.
Some raisins with her, and that's what she's.
All right.
Yeah.
like a lot of things before you do it, right? Second nature.
Yeah.
Then we'll send you off anyway.
Absolutely. Very excited. For us.
What's going on in your life?
Oh, yeah.
Yeah, absolutely. Yeah.
Oh, you mean longer trips? The first half of the slides are really more.
Do you need me to roll some heads?
No. My secret where I really want to spend most of the time.
Oh, right.
It'll be a challenge to think of an hour.
So you get 50 miles.
Nice. 4 miles in and out.
I was looking at CA this morning,
And I.
Oh, well.
and 3 of them
Salmon spread.
don't pay me insurance at all.
Good. That would have been good.
Oh, now it's not very Come on.
You gotta see fresh window and the executive suite.
Did you ride your bike today?
I actually did.
You look like you're in your bike riding.
Yeah, well, I'll be biking a little later. Raining a little bit. I know, our dog is all over our bed, jumps up and down to half an hour, catch and catch. Yeah, later that morning.
Do you have a job yet? Sorry?
Tony and I were together.
You should have a timer.
Frank, he's from his backyard. Well, here, you gotta get these bushes. They're only like $8.
Pascal sauce.
Here in town. Oh,
Paul was taking over the whole area. So they got the birds.
Now put it in the newsletter. I don't even know. How are
That's right, it's always a little about like, yeah.
This way I have some stuff. I didn't want any samples in work.
Way out there doing season anymore, but I can't put it a big roadie and well. Could be worse.
Really? There's more to go.
That or get into the next week.
Alright, so whatever works.
Okay.
I don't need to get a signature.
Okay.
Yeah, that's right.
I just got owl place.
And I guess it wasn't.
Private place. What was it, Mark? I had one else.
Yeah, I have it.
Year to 2000.
Is that your secret to aging, Gary, is washing down your coffee with Coke?
Oh, really? That's not right, but I would. Yeah.
Sometimes any caffeine is so
You have met. You have met.
Are you going to do a tour?
20 years.
Does Carol have any?
Does tour have a
Let's go ahead and get started.
Just two quick announcements. Again, the last session of the year is the 22nd on the Wednesday, and inadvertently there's a conflict, but I'm not changing that date again. There's a talk up at Children's on eosinophilic esophagitis that evening that we're not sponsoring. I don't actually know who set that talk up, but a couple of people pointed that out to me. But at this point, after all the emails I sent out voting on different dates, it is what it is. So there is a conflict, and if you want to go to the talk at Children's and then come over to the end of our session, I think that'll work because you can just walk from one to the other. The other thing is, later in the year, I'm going to put together the directory for our allergy division as I did last year. So if there are any updates that anything was wrong with your own listing, or you have a new colleague or anybody else who should be listed in there, for anybody on the line who's listening anywhere in the region, send me an update. Frank is going to talk this morning. I'm always confused by this topic, so I'm very happy he's going to straighten it out, and I can explain this every day to patients. Why do some patients with IgE to foods, in other words, in particular positive skin tests, tolerate these foods? I need to know this answer, Frank.
All right, we have 1 hour.
Right.
Let's see if we can get to the bottom of this. For the purposes of this talk, really no applicable disclosures, although ironically this last week we've actually started a epicutaneous peanut desensitization study. I'm not going to be plugging that or anything. So why do some patients with IgE to foods tolerate these foods? Well, I thought about this a little bit, and my first response is you have to sort of have a Sherlock Holmes approach. And I apologize for maybe Len and Nick, who are expecting Basil Rathbone instead of Robert Downey Jr. And this is not something that, when we're evaluating patients necessarily would come up. But if you're seeing patients that have been evaluated elsewhere, let's say, it's always good to think about other possible reasons why the patient might think that they're allergic to the food, and in fact, they may not even have IgE to the food. And we're all used to this, obviously, patients that have dermatograpism when they're skin tested, or alternatively, if they've had immunocaps done, there really is some evidence that if total IgE is over 1,000, you do start to get background noise in immunocaps even. And then the middle issue is probably a little less of an issue because of how extracts are now prepared. But actually, if extracts do not undergo dialysation, you can actually get histamine and other vasoactive amines that can add to positive skin tests. And potentially, ironically, when we're all trying to use fresh fruits and vegetables to skin test to avoid potentially false negatives, there's a slight chance that some of these foods actually might be irritating to the skin, and you could actually get a false positive. So I'm not going to belabor this too much. And for the first part of the talk, I really want to just throw out some informational things here. I want to spend the bulk of the time actually talking about tolerance. So I'd appreciate maybe if you'd hold questions on this portion until we get farther into it. This is just to show you a study about 20 years ago done at National Jewish in North Carolina, where they actually showed that a very high percentage of extracts, if they are not dialyzed, actually do contain vasoactive amines. Now, if you look at this carefully, this is order of magnitude smaller than what we use for a histamine control. But the theory goes it may, turn a borderline skin test into something that somebody would call positive. Again, I'm not going to go into this into detail, but this is actually, and if you have time, it's actually worth going on the FDA site. It's actually a very nice compilation of all sorts of adverse reactions to foods, food allergy, and so forth, and some nice references. But they make the point here also that if total IgE is above 1,000, you have to need to be very careful about interpreting low-level positives. Interestingly enough, there was no reference attached to that comment, however. And then more recently, this was actually an abstract at the recent WAO meeting in India, a study done in National Jewish, and just to cut to the bottom line, demonstrating in patients that, again, if total IgE is over 1,000 and you then start diluting, you actually don't get serial dilutions of what appears to be positive reactivity to foods. And the author's conclusion here is that you need to be very careful, and in fact they actually recommend diluting serum down at least fourfold if the total IgE is above 1,000 until you would get the effective total IgE under 1,000 to actually give a better sense of potentially true positives. All right, so that was my Sherlock Holmes portion. The more likely group of answers are, and I put several researchers, most of which you'll recognize, Hugh Sampson, this is Paul Boyce at Northwestern, Wesley Burks, and Gideon Lack. Not that they're the oracles on the subject necessarily, but I think they would give you some of these answers, that there are actually epitopes in the ingested food that are different than those that are being tested. And we'll spend a few minutes just briefly going over what's been described in the literature in terms of the effect of specifically heating and also digestion on these structures. Another theory is that there may be a relatively low density of the epitope or structures of the epitopes in the food that reduce the likelihood of cross-linking, or what you're picking up in a skin test is a slightly different protein than what's actually the primary problem, for example, birch pollen. The other theory, is that perhaps there's a disconnect between what we observe in the serum or skin testing and what's actually going on in the GI tract. I would submit that that's fairly rare, but it's certainly a possibility. Another option is that the patient actually has acquired natural tolerance, something that, for example, we know almost always happens in patients who are allergic to egg and milk, and rarely happens, for example, in patients who are peanut or tree nut allergic. And then the other possibility is that in a study situation where we've desensitized the patient, we've actually created tolerance. So in this next section, I'm going to very briefly talk about several antigens, just because I thought you'd find it interesting. Some of us are aware of this already, but I learned a few things about some specific antigens that are worth keeping in mind when we're evaluating patients. So this is looking at the effect of heat on antigens. Wheat is generally obviously eaten, cooked, or heat-treated, and obviously patients clinically still have trouble, and it's consistent with the fact that most of the allergens are relatively thermostable. Egg, as we all know, clearly if you cook egg, some of the antigens, but not all of the antigens, will sort of become less relevant, and it turns out that ovomucoid is the one that's heat-resistant. Ovalbumin is relatively labile. And a number of studies have shown that if you take, for example, a child who's egg allergic, or take a group of them, 70 to 80% of them can tolerate a sixth of an egg in baked goods. And the key point the authors make there is what they call wheat matrix, or essentially flour. And essentially the thought is that what we're doing, if the patient's allergic to ovalbumin, is we're denaturing that antigen, but we're also spreading out the ovomucoid, which they also could be sensitive to, and reduces the likelihood of cross-linking and mediator release. Milk, a somewhat similar story. Of interest, the process of pasteurization really causes very little change in allergenicity. High treatment with heat for a much more extended period of time destroys most of the whey, but most of the casein antigens are retained. So the data, again, are very similar. If you take milk allergic patients, about 70 to 80% can tolerate it in baked goods in a wheat matrix. And the theory, again, very analogous to what happens with egg. In fruit, and Dr. Park gave a great talk about several of these antigens a year ago, there are several proteins which can cross-react, for example, with pollens. The PR10 proteins, and an example is BET-V1, is very heat sensitive. The profilins are also quite heat sensitive, and BET-V2 is a good example of this. Whereas a third group of proteins, the lipid transfer proteins, are relatively insensitive. So if that is a relevant allergen, it's much more likely to cause clinical symptoms. And keep this in mind in a minute. We'll talk about the effect of heating, which is also relevant for some of these antigens. The phenomenon of enzymatic digestion in the stomach, which is also relevant for some of these allergens. And as we'll see, may explain why if you eat a raw fruit and you get oral allergy, actually pepsin very readily degrades these antigens, so often it eliminates the risk for more systemic reactions. Parvalbumin, quite stable. In fact, the authors note that, interestingly enough, some patients will tolerate raw fish but actually react when it's cooked, sort of the opposite of egg and milk. Shellfish is minimally affected by heating, and in fact, sort of analogous to peanut, boiling or steaming shrimp may increase the size of a reaction. Tree nuts, roasting may reduce but doesn't abolish allergenicity. For example, with hazelnut, there's not a lot of data with most of the tree nuts, but the theory goes that heating it's unlikely to destroy their allergenicity because they're fairly rigid structures. Sesame story is similar to peanut, little effect of heat. This is, and again, we've all heard this time and time again, that roasting peanuts appears to increase the risk for allergenicity compared to boiling or frying. And you can get a little glimmer. I know it's probably a little hard to see maybe there, but essentially what you're looking at here is serum containing IgE to peanut, and looking at the ability of relevant antigens to absorb that or inhibit that. You can see that when you roast it, you get much more inhibition of the IgE that's present in the sample. Another interesting thing is something called the Maillard reaction, so that if peanut is around with sugars, and especially if it's heated, it creates this cross-linking phenomenon that makes it much more likely to not be degraded. Soy, again, very high heat for an hour does not completely eliminate, although it partially destroys some of the allergens. Another interesting comment, we all traditionally, in patients that have some reactivity to soy, often feel that soy lecithin isn't a problem, and that's typically true, but the authors point that depending on the product, there's a wide range of soy lecithin in certain products, and this could be relevant for some patients. Analogous to a lot of the oils, again, if soy is distilled, there is very little allergenicity. Celery is kind of an interesting phenomenon. Again, parts of the celery phenomenon are due to profilins, but if you actually have IgE to a cross-reactive carbohydrate determinant in celery, you can boil this essentially for 60 minutes and it's still allergenic. maybe the source of all those patients in Boston that have exercise anaphylaxis. Lentils, also kind of interesting. They actually may be more allergenic when cooked, much like peanuts. And really, this is just a study showing that if you actually boil the extract, you get much more improved specificity and sensitivity versus using standard extracts. And I think the take-home message here is, if possible, when you're skin testing patients, obviously use the food in the form that they appeared to react to if you want to get more sensitive and specific results. Mustard, again, relatively resistant to heat. I think we've all seen a few of these patients over our careers. It's certainly a rare allergy, but it can be quite impressive. Often the patients that anaphylax are reacting to a lipid transfer protein in mustard, and the point of this study is to show you that it often relates to cross-reactivity to mugwort, and if patients have that, they also tend to react to similar vegetables, cabbage, cauliflower, and broccoli. All right, well, let me say a few words about the effect of digestion. Another thing to keep in mind when we're trying to explain away why does this patient have a positive skin or blood test, but they tolerate the food. This is kind of an interesting group of studies where they actually produced a simulated gastric fluid, and you can see the constituents over on the right here. And the columns are, for some reason the arrow is not coming up here. There we go. So what you're seeing in the columns here are the scenario in a so-called fasting patient. And keep in mind that once you start eating and obviously you use up acid, you actually get a slightly different milieu. And this is over the course of 15, 30, and 60 minutes. And so these authors looked in vitro, using this as a proxy for gastric fluids, the effect on various antigens. And what you can see is that in contrast to some other plant proteins that are literally toast when you put them in gastric fluid, within seconds, several of the relevant allergens that we see in patients are quite resistant. This is an hour here. Some are relatively less resistant, although as we'll see, what we're looking at here is just that the protein has been broken up at all. And that doesn't mean that you still don't have chunks of the protein that are allergenic. And the authors kind of make that point here. But you can see that several of these antigens are relatively resistant. And if you look closer at this, again, another group of plant proteins or plant allergens that are relatively resistant to degradation, these two families, the prolamine and cupin superfamily, and specifically look at some of these antigens, you can see that by and large, although there's a couple of exceptions, these are quite resistant to degradation. And then if you look at the more sensitive allergens, so that ironically the thermostable antigens in egg and milk are relatively, at least partially, sensitive to degradation in the stomach, but the point is that it still leaves big chunks, and unfortunately for many patients these are still relevant allergens. Tropomyosin in shrimp and ARA-H1, for example, in peanut. And again, this just shows you that, again, that these ones that are more pepsin sensitive, but as I mentioned, it doesn't mean it renders them non-allergenic. Highly sensitive allergens, we talked a few minutes about the effect, again, of heat on PR proteins and profilins. So, again, a reason why when patients eat these raw, they're potentially an issue in the mouth, but these are very readily digested in the stomach, so it's very unusual, unless perhaps you add in exercise to increase absorption of the antigen, for them to be a clinical problem. And again, just showing you that data. See how we're doing here.
Frank,
All
how
right.
about salivary protein digestion? How much of that occurs, do you know?
I didn't come across that, but I think the general feeling, just in terms of unless you're literally like you're chewing gum, it's not there long enough to have a huge effect. That's a good question. All right, so as I mentioned, I'd like to spend the rest of the time talking about the last two potential kind of reasons for why a patient might appear to have IgE to a food and actually tolerate the food.
Frank, before you move on, with that information, especially what's either enzymatically or heat or stable, how effective do you think the new component resolution testing would be to tell us more specifically what IgE people have, the specific proteins?
Well, I think it can be quite useful in terms of, granted, it's literally, here's a result, what is the risk for that patient, to have a systemic reaction. And part of it really, I think, is just based on, if you do the study, that's the result. But some of this data, that these antigens are relatively thermostable or, pepsin stable, as opposed to, for example, in peanut, the ones that are more plant proteins that have a lower risk. So I think it could be quite useful. You could think of it also in, if a given patient's curious, I suppose the hazelnut story or the shrimp story, actually see what antigens are relevant.
For those of you who do a lot of food allergy, you've been using that test? No, it's expensive.
Yeah, I've done it, I think, maybe only about 8 or 10 times now, and typically the sort of classic scenario, again, for a lot of these patients, is it was picked up when they were tested when they were 1 because they reacted to egg or something else, and somebody did the 5 most common antigens, and they've just avoided it ever since. The other proxy that I've sort of tended to do, let's say, and it often comes up in a, junior or senior in high school, and the parents worried about them going off to college or something. Now they're suddenly worried. Are they allergic to things that would potentially explain this? In other words, are they allergic to alder, birch, or grass? And if they are, well, it still doesn't answer the question. If they aren't, it suggests to me that they're really not going to have cross-reactivity with most of these lower-risk plant proteins. And it's, I think, sort of a cheap way of maybe avoiding doing the test. But I've done it, and literally half of these patients, it's actually led to challenging them. And these are, just to put it in perspective, patients that have, 6-7 millimeter wheals to peanuts still, or IgEs of, even 8 to 10.
So did you challenge them if they were like this era 8 positive? Is that what you did?
Correct. Right.
And they were 2 and 1 negative.
Correct. And 3 negative, yeah. Exactly.
Are any of the commercial extracts prepared? So like peanut is done with dry roasted peanuts for the extract instead of...
Not that I'm aware of.
Because it would think that if it's that much different the way you talk, they would think that the manufacturers might look at that and say, hey,
yeah,
we're going to get better answers.
No, that is a good question. And I also, just back to the very first thing, which is a minor thing, I don't know to what extent they dialyze all the extracts now. And I know that the fellows are going to go to Hollister Steer in 6 weeks, so they'll give us a report on that. It should be interesting. They're going to go through the plant over there and do a little botany walk. All right, well, this gets to be a little overwhelming, but bear with me here. So I'm going to sort of lead you through, and this is an excellent article, by the way, kind of summarizing a lot of this. First, some background stuff, kind of bits and pieces of studies, and then trying to put it all together about what we think happens with tolerance. And these authors have sort of divided this into what they call inductive sites, the gut-associated lymphoid tissues, including Peyer's patches and local lymph nodes, and then effector sites where this process begins in the lamina propria and epithelia. The gut-associated lymphoid tissues are, not surprisingly, critical for this process. It turns out in these lymphoid tissues there are these somewhat specialized cells called M or microfold cells that can actually actively transport material from the gut lumen to the lymphoid areas for immune processing. You know, it's unclear if they directly otherwise play a role in tolerance. But if you can actually, in animal models, deliver the allergen right to these cells, it greatly facilitates tolerance induction. Lamina propria dendritic cells are also very critical for the induction of tolerance. And to this point, if you orally administer antigen, sorry for the typo, is detected in the gut epithelia and lamina propria minutes after feeding. Antigen-loaded dendritic cells and macrophages can be observed in the lamina propria, and again in a relatively short period of time after feeding, for example, dextran or albumin. So how does the antigen get to the dendritic cells? Well, it turns out it's felt there's sort of four pathways. One is that small peptides can actually get through tight junctions. Larger proteins are transcytosed with fluid as fluid is absorbed. And then there's these little pockets of partial proteins and MHC components from the edge of epithelial cells that are actually turned into an exosome, and these are actually presented to the dendritic cells. And then also some evidence that these particular type of macrophages essentially have these long arms that reach into the lumen and grab it and can present it to the dendritic cell. And this is just a cartoon showing you, for example, in a Peyer's patch, these M cells processing antigen, and then the various mechanisms, the tight junction, transcytosis here, the exosomes containing antigen and MHC, and then here's one of those long-armed macrophages, ultimately presenting it to the key player, which are these dendritic cells. The other question is, well, how does this end up being actually a systemic process? I mean, that's all fine and good, what happens in the GI tract. And interestingly enough, at least portions of food protein can be detected in the blood after eating food. We see upregulation of T cells in local lymph nodes. And of interest, you can actually take serum from protein-fed mice and can induce specific tolerance in naive recipients, suggesting that this truly does become a systemic process. How and where does antigen get absorbed that helps contribute to this process? It's felt that the liver is a key player in this, and the authors talk about, obviously, the blood supply to the liver. Interestingly enough, if you inject in animals antigen directly into the portal vein, you get tolerance. If you divert blood away from the liver, it largely prevents oral tolerance. And nobody knows for sure, but it's described that various antigen-presenting cells in the liver, including these sinusoidal endothelial cells, Kupffer cells, and what are called plasmacytoid dendritic cells, and there's buckets of them in the liver, all of them tend to favor tolerance. So it's a very tolerogenic organ, if you will. And then some of this process also happens probably in peripheral lymphoid tissue and in the spleen as well. And this is just a cartoon sort of demonstrating what we just talked about, how the process happens. So It turns out another important part of the piece is transporting this lamina propria antigen to the local lymph nodes. This is critical for inducing at least systemic tolerance. It turns out it very much requires the chemokine CCR7, and that's been proven by taking genetically deficient mice. You do not get tolerance. And similarly, not surprisingly, it requires these local lymph nodes, which if you either destroy them with a lymphotoxin or remove them, again, you don't get oral tolerance. So what are these sort of specialized dendritic cells that are so important? Well, they specifically have a marker called CD103. I'm not sure that anyone needs to know this, but it actually is part of the integrin chain, or equal to the integrin chain alpha. It should be alpha E. They stay in the lymphoid, the mesenteric lymph node area. They do not leave there. Another interesting part of this process is, and a critical role apparently, is that they metabolize dietary vitamin A to retinoic acid. And it turns out that retinoic acid is very important for inducing gut homing, which it does by producing more CCR9 and also upregulating another integrin on T cells. And so interestingly enough, gut homing is blocked by antagonists of retinoic acid synthesis. And it sort of made me think about, is there any role for, we talk a lot about vitamin D. Is there a role for vitamin A, perhaps when you're trying to induce tolerance? I don't know that anyone's looked at that. And then this process also induces T regulatory responses. And it does this, and part of this is also related to retinoic acid. It activates something called transforming growth factor beta, and again, upregulates yet again another integrin. And these are important cofactors for sort of telling naive T cells to turn into T regulatory cells. We've all heard many times about the FOXP3 positive T regulatory cells, and it's certainly felt that at least somewhere in the process they do play an important role, although as we'll see in a minute in a study with milk, it may be a transient role actually rather than a permanent role in inducing tolerance. As we've just talked about, these CD103 positive dendritic cells and local lymph nodes are crucial, as are the availability of vitamin A and other structures. If you take such cells in a tolerant mouse, for example, you can transfer oral tolerance to naive animals. If you deplete these cells, you remove tolerance. And the short story is, it turns out these are actually inducible Tregs versus natural Tregs in the thymus. So piecing this all together, the model here is once these dendritic cells have antigen, they migrate with it from the lamina propria to the local lymph nodes. In the local lymph nodes, retinoic acid produced in these dendritic cells plus TSLP help induce critical integrins and CCR9 on antigen-specific T cells, which again are part of the gut homing process. And then we already talked about how retinoic acid plays a role in induction of inducible Tregs. Clonal expansion of these Tregs is driven by IL-10, although it's thought that some of that process is also driven by a different group of T regulatory cells, the so-called TR1 cells, and also IL-10 that's produced by the macrophages. And then it's felt that these T regulatory cells may also exit the area and somehow promote systemic tolerance also. I don't know how well you can.
Frank, I assume,
Yeah.
so that's sort of a complex story why most people are not allergic to things.
Right.
That we're set up to tolerate foods. That's a smart way to be made. Obviously, people who are atopic, something's wrong in that line of thought.
Well, we'll talk about that. At least briefly in a little cartoon, sort of that, again, dichotomy or balance. But something to think about, which is still not well understood, is, why do most people outgrow egg and milk allergy and not peanut and tree nut allergy? So this, just very briefly, let's see how we're doing here. I'll show you a few separate things. In other words, the question is, the theory goes that allergy in general might be due to a specific lack of T regulatory cells for each antigen that a patient's allergic to. And that's sort of an indirect inference because we know that that's low in such patients. And for example, at least with traditional immunotherapy, you can demonstrate that such T regs actually increase. So for example, for grass or dust mite or cat, well, is that also relevant for the food story? And I think that there probably is ultimately some evidence for that. But a couple side points. So this is looking at peanut allergic patients versus previously peanut allergic patients versus controls. And just to show you in brief what we're looking at here, essentially this is the ratio of Th1 type cytokines to Th2. So if we're looking at presumably more of a Th1 response, that number gets higher. So for example, in peanut allergic patients, you can see that that is a very low ratio, suggesting more of a Th2 type response, not a surprise, in contrast to the response to other antigens that the patient's not allergic to. Interestingly enough, which was kind of a news to me, even though it's not necessarily an IgE producing response, it turns out tetanus toxoid tends to favor a Th2 type response. So that's why the authors put this in as a proxy for a Th2 type response. They didn't really explain that much, but that's why they put it in here to kind of put it in perspective. Now if you come over here and of course look at a patient who isn't allergic, not surprisingly that patient makes more of a Th1 type response to peanut. And then if you go to the far side and compare peanut allergic versus not allergic versus those that were peanut allergic, the 1 out of 5 that outgrow it, they look very much like a patient who's literally never been allergic. Now this, I just thought I'd throw in, you'll see some suspicious authors here. This is Kelly Hetherington, Dave Robinson, and Bill Kwok. And I know Fatima and others, David John, for example, has also worked with Bill. This doesn't directly relate to the question, but just to show you the idea that if you actually complex MHC with a relevant, not only a full antigen, but actually a short amino acid sequence and create these tetramers that can light up in a flow situation, you can actually demonstrate very specific clones that are produced by these patients and actually clarify the actual part of a particular antigen that the patient is responding to. So it actually gives you a very powerful tool to very specifically look at, for example, the process of tolerance. Now this was kind of an interesting study, and I think there's at least one surprise in it. Study of looking at, and this is an in vitro study in milk allergic patients, shorter study for 7 days, and essentially looking at the induction of T regulatory cells, comparing milk allergic patients that cannot tolerate milk in any form, controls, and then HM are those that can tolerate or pass a heated milk challenge. You can see that they have induced T regulatory responses. Of interest, though, if you then look a month or two later and compare, so this is on the right here, is looking at patients that can tolerate heated milk, and then after the challenge, somebody said, well, I still don't want you to eat it, so don't eat it. If you look at the ones that are actually regularly ingesting it, which we all feel helps induce more tolerance, of interest, their T regulatory cells actually decreased. The authors really didn't have a great explanation for this, but suggesting that maybe somewhere along the line a second process happens once you start getting a T regulatory response that is actually more important for actually shutting down IgE production. And they speculate perhaps a different group of T regulatory cells, like the TR1 cells, might be the explanation. But personally, I would have expected just the opposite, that if you're ingesting it, you might induce more of these T regulatory cells. So here's a cartoon, back at least partially to Len's question. And I'll start over here, sort of looking at the allergic state. And obviously you have to have the genetics for this as well. But the theory is that if you have antigen in these dendritic cells and the presence of OX40 ligand, and it turns out that TSLP is an important adjuvant factor here, it basically tells these T cells to create a Th2 phenotype and also downregulates inducible T regulatory cell formation. Whereas in the patient where you're developing tolerance, you get the other pathway we talked about, where in the presence of retinoic acid and transforming growth factor beta and a few other structures, you actually induce T regulatory cell responses. The other, I'll just mention, we'll mention this in a minute, it turns out that we talked about the microbiome a fair amount, and we've already talked about the role for vitamin A. But as you can see in the top of the slide here, it turns out that Clostridia species and something called polysaccharide antigen on B frag are actually very tolerogenic species. So they help induce tolerance. So another question comes up, does tolerance actually naturally occur? And if so, how does that happen? We've already talked about the clinical reality that egg and milk almost always go away. Peanut and tree nuts rarely do. So I'll show you a couple of studies. And notably absent from these studies are peanut studies, which are still a work in progress. This is an interesting study of looking at either egg or milk oral immunotherapy, and you can see the doses that were used for a period of 21 months. And then at the end of the study, somewhat arbitrarily, the authors waited 2 months and produced a double-blind placebo-controlled challenge, and they were all excited by the fact that 36% of the patients had induced tolerance. But then somebody said, wait a minute, let's test the ones that didn't have immunotherapy, and it turns out the rate of tolerance is identical. So it sort of begs the question with this sort of immunotherapy, although the theory is that if a patient, for example, can tolerate egg or milk and baked good, that you may speed up the development of tolerance. It sort of becomes questionable in the big picture how much difference that actually makes. Now this is a sort of complicated study with milk, but the idea here was comparing slit with 7 milligrams versus, as you can see, 1 or 2 grams orally, and then at various times in the study, these patients were challenged with, I think, what all of us would agree is a reasonable, 8 grams of milk. If you pass that, you're probably pretty tolerant. And then at the end of the study, again, they waited longer, actually 6 weeks. And you can see that there was a spectrum here, kind of in favor of the higher dose you use, perhaps you're more likely to induce tolerance. Now, in this study, they again did not test the placebo groups. So we sort of don't know the background answer in that time frame. That's another interesting study from, you can see all of the major kind of U.S. food allergy researchers, slightly different groups as it turns out if you look at specific IgE and skin tests, but probably same ballpark. And then a placebo group, oral challenges at various times to see after 10 months what percentage had developed more tolerance, and even if they hadn't, they stayed in the study and were again re-challenged at 22 months, and they felt that they could desensitize about 75% of these patients. But again, after 2 months, only about 28%, so that sort of magical 30% or so seems to be what you end up getting with tolerance, which at least in that first study seems to naturally happen, even if you don't do such therapy. And again, importantly in this study, the placebo group was not re-challenged. And I won't go over this into detail, but if you look at those that sort of passed at 10 months, it turns out what correlated with tolerance at the end was the presence of specific IgG4. If you only passed at 22 months, the most reliable predictor of tolerance was the size of the skin test. And you can see, actually, not surprisingly, all of the ones that passed had no positive skin test. So not a surprise. So if you look at all of the studies so far, the only consistent finding that seems to correlate with tolerance is the presence of specific IgG4, again, not dissimilar to what we see with traditional immunotherapy for inhalant allergens. Still, there's a lot of studying to be done to see kind of how this all might happen. And so, kind of related to the milk study I showed you a few minutes ago, the question is how much of this is due to the induction of a T regulatory cell response, and ultimately, is there some separate process that actually decreases allergenic sensitization? In other words, the other side of the equation here. And I don't think anybody really knows. So let me say a few words about potentially future types of immunotherapy. And these are just some observations that if you feed antigen to naive mice, you can actually induce T regulatory response. And the key point there, which probably applies to people, and these are mice that are genetically programmed to make an IgE response, it's how you do it. In other words, the level of antigen needs to be very low and over a sustained period of time and gradually built up. And obviously, as opposed to, if you will, a normal amount of the antigen, that will actually create an IgE response. And that's very analogous to what we see with micrograms to milligrams of injected immunotherapy for pollens versus, of course, what happens with natural exposure. Similar to some of the human data here, egg-sensitized mice given OIT are desensitized, but as a group tend to not develop tolerance. In other words, if you then stop that for a period of a couple weeks even, very few of them actually have tolerance. And why that is not well understood. And then another observation is that if you sort of bypass some of the things that might otherwise happen in the GI tract and actually go directly to the liver and spleen, you can actually produce tolerance. Obviously not a practical therapy in people, but an interesting observation. Another consideration is fiddling with the allergens. At least at a minimum, to reduce the risk for bad reactions in this process, and possibly to favor the development of tolerance. These are just some thoughts and some things that are being looked at. One is partially degrading the allergen, much like, again, the effect of cooking or what might happen naturally, but still retaining enough of the antigen to induce tolerance. It's been described that if you add mannose to the antigen in various forms, you can help induce these TR1 cells and produce IL-10, which downregulates the process. Another approach is, again, bypassing the GI tract, and literally last week, I think Steve Tillis was involved with doing some challenges in a study that we've just started, comparing placebo versus actually I think 3 forms, 50, 100, and 250 micrograms delivered epicutaneously in a patch to see if you can induce tolerance. And I believe at the end of that study, in addition to proving that the patient's relatively less sensitive, ultimately we'll see at what level they've induced tolerance. Another approach is, again, for example, peanut being an example, taking these so-called high-risk epitopes that some patients are sensitive to, actually modifying the epitope and presenting this, for example, intrarectally in mice, you get induction of tolerance. There are apparently human phase 1 trials, although it wasn't clear. Presumably it's not intrarectal in people. It's unclear how these are being delivered, possibly orally. But then the question comes, of course, protecting them from degradation could be an issue. Another study, and this was a study that got a lot of splash from the Northwestern group, is the sort of clever idea of taking cells, if you will, self-antigen, and coupling it with antigen. You can actually induce tolerance in animal models. And there are now some trials looking at this in autoimmunity, but not necessarily allergy yet. As I mentioned a few minutes ago, there are these adjuvant factors in the sense of helping to induce tolerance in various microbes that might have some clinical utility. And I thought you'd find this interesting. This was a study that many of you have probably seen, was just published a couple months ago. A fairly clever idea of actually coupling a relevant allergen to the bottom of IgG, and I'll show you how that works here. So this is such a molecule. So this is the bottom part of IgG that binds to its receptor with antigen, which does 2 things. It helps potentially block or take up some of the IgE receptor sites for the antigen, but more importantly, through inhibitory pathways, downregulates SYK, which is an important part of the pathway leading to degranulation. So it's kind of a clever way of sort of downregulating the process. And I'll show you, just very briefly cut to the chase here, to show you, here's an example of looking at the response to antigen, and you can see that high-dosed injection with this fusion protein looks a lot like, not totally, but a lot like control, and you can see that you get this blunted inflammatory response. And if you look at, if you will, a proxy for histamine response in the skin, here is the treated mouse versus various doses of mouses that are probably headed towards stage 5 and dying in the study. So another interesting possibility. So this slide just summarizes the various ways that we might be able to sort of have a new ways of encouraging tolerance in immunotherapy. So to summarize, the answer to the question, why do some patients with IgE to foods tolerate the foods? Always keep in mind that the patient might not really have IgE to the food. And we talked about some possible explanations. Again, the idea that epitopes in the ingested food are different than those being tested. Another thing to keep in mind, of course, as you think about this, is the opposite problem. Why does the patient not appear to have IgE to the food, but they actually react to the food? Is just the opposite thing, that we're actually not testing for the relevant epitopes because of perhaps the way the food was prepared. The other theory is that there's a relatively low density or perhaps a slightly different structure of the epitope in the food that reduces the likelihood of cross-linking. And an example here would be, again, many of the plant proteins that are relevant for oral allergy syndrome and the effect of theoretically spreading the antigen out. We talked about the potential disconnect. I think that's probably rare. And then the phenomenon of natural tolerance, which, obviously I don't think anyone's come up with a great explanation. If you have a baby who's allergic to egg and milk, and let's say for whatever reason they never get a baked egg challenge or something, and eventually they outgrow it. Well, why does that happen? And why doesn't it happen if you take another child that's allergic to peanut? And then finally, of course, potentially inducing tolerance with a study protocol. And I'll leave you with a question mark, at least as it relates to the egg data. Does that really happen? Or are we just fooling ourselves and keeping the patient busy, and that would have happened anyway? All right, let me stop there.
Frank, we heard a couple weeks ago about the interesting way to desensitize people by injecting allergen into their inguinal lymphatics. Then you said you could desensitize mice with intraperitoneal.
Yep.
If you have mice that are sensitive to peanut and you inject peanut protein into a mesenteric lymphatic, or can you desensitize them as well?
Yeah, I would think so. It seems pretty clear that if you hopefully avoid an end organ that's going to, stimulate sort of the wrong part of the pathway, that you probably could induce tolerance. So, and I'm not aware of any...
No human studies of inguinal lymph node injection of peanut?
No. There were some. I think we talked a few months ago about some intrarectal studies in Germany that they were actually doing.
I was just impressed that the guy said you could desensitize people with 2 shots if you give it in the inguinal lymphatics. I just wondered if we could do foods the same way.
Yeah, you would think so from the mouse data. Yeah.
So it seems to me, we don't know whether these techniques that are being used now result in long-lasting tolerance. But even if they don't, there's probably ways of conveniently sort of continuously exposing them without the idea of anaphylaxis. Like, for example, I have no idea whether this transcutaneous immunotherapy is going to work, but if it does, people don't need epinephrine on this stuff. They just don't have systemic reactions, the several hundred that have been treated with it for months. So as opposed to the oral immunotherapy where that's a problem, and if you have to be on it indefinitely, it's like, well, you could do the math with the statistics, but eventually somebody's going to have a really bad reaction.
Right.
So I don't know what the answer is, but...
Well, the other intriguing thing, if you believe, I honestly haven't seen this myself, but if you believe the data from Bob Wood and others about, 10 to 15% of patients with peanut allergy, if they don't keep consuming it after they're okay,
right,
they seem to get the allergy back. It's sort of and yet I've certainly never seen that with milk or egg. Although presumably everybody has a few cupcakes a week or something, or cookies, so that might be a little different. But it's sort of, is there something intrinsically different about some of these antigens that you can get the T regulatory response either naturally or with immunotherapy, but is there something else kind of alluded to, like with the milk, that starts happening that really shuts off almost the gene, if you will? In other words, you stop making IgE, so it's never a problem, even if you didn't eat it for 20 years, where it clearly is with peanut.
Frank, there's sort of different stories here, but
Yeah.
it would seem to me, obviously, people who are not genetically allergic, they're set up, when they eat a food, they naturally become tolerant, and people who are genetically allergic are different. So, I mean, the first question is, if you still truly have IgE, and you don't react to the food, what's stopping that from happening? The only thing that I could think of is you have IgG subclass 4 that you may become naturally tolerant and you block the interaction of the IgE. So I guess the question is, if you become tolerant, does tolerance mean that you stop making IgE, or that you make enough blocking antibody or some other blocking mechanism that you can still live with your IgE, but it doesn't cause disease? That's the way I think of inhalant immunotherapy.
And I don't think anybody knows, but it makes you wonder if maybe there are two different scenarios where, well, like you said, if the patient still has IgE, that tells you they're still producing IgE to at least some component of the food. But I think with some of these antigens, eventually, like with egg and milk, their IgE goes to 0, which might be different than the peanut tolerant patient, where they have this sort of thing that prevents it from being a worsening clinical problem. But if they stop ingesting it and stop making the IgG4 and the Tregs, they might revert to where they were.
So when you measure like tolerance mechanism, I mean, other than clinically knowing you're tolerant, if you measure those outcomes, do Treg cells stop you from making IgE, or do they help you make tolerogenic mechanisms so the IgE is no longer a clinical problem?
Yeah, I mean, the theory is ultimately it should stop you from making IgE. The question in some of these studies is, I think, is it long enough? And the answer is, I think, pretty clearly no. In other words, it's fairly easy to desensitize 80 to 90% of patients, but actually inducing tolerance in as little as 2 months later clearly isn't happening very much.
I mean, we know with inhalant immunotherapy, people make more IgE when we start to immunize them.
Right.
They make blocking mechanisms so that heightened or remaining IgE is not clinically significant.
Yeah.
There's an outside question here from Chuck Jackson. What about the infancy window of tolerance to food exposure? Will the AAP guidelines for withholding peanut until age 2 be changed?
Yeah, I think the answer is yes. I mean, I think it's clear that's clearly a work in progress, and if you've ever worked in the AAP, it involves about 12 layers of committees, including the Committee on Committees. In an active Congress. But, I think clearly there is, which we didn't talk about, but there is some evidence that we're actually probably the best time to induce tolerance may be the very time where we tell parents, or we have for a generation, not to give these antigens if their kid has eczema and their sibling has a food allergy, is actually from 6 to 24 months. So I think that the million-dollar question, though, is, and since we don't have the genetics, in other words, you can't do cord blood and say, I see, yes, this child's destined to be allergic to egg, but it's probably how you introduce those foods. For example, like the teething biscuits in Israel, when you think about it, it's, as kids are developing teeth, it's probably micrograms of antigen. So we're not talking, 10 grams of the food initially, but the level of antigen may be critical in terms of going the tolerant way versus producing IgE. And it's hard to study, like the LEAP study, as you know, because we don't have the genetics. You have to take these high-risk group of patients and wait 5 years until they're in kindergarten to answer the question, did the teething biscuits really reduce the expected frequency of peanut allergy or not? It's pretty laborious, but again, still a lot of mysteries.
getting back to the idea of IgG4 tolerance and higher levels and seeing, our learned colleagues in the alternative community would say that person has then become allergic based on their testing. And I think it's very analogous, most likely, then to the inhalant immunotherapy, where we're seeing this blocking effect. But stepping back, and this may be more of a teleologic argument, but my idea is that the gut is sampling. It's been pathologized, or trending to pathology in some circles, where this leaky gut is causing all these problems. But maybe the gut is sampling at this young age. The gates are wide open and it's being exposed to the immune system, so to speak. You tag it with an IgG molecule and say, we've been there, we know this. We look at babies put everything in their mouth. Sometimes I wonder if they're just sampling their outside environment, just showing their body, this is the whole world, let's not get excited about this.
Right.
So, anyway, there's some things I think about with this. And I wonder, like we said, the AEP, we went the exact wrong direction in saying, let's not give kids these things. When in fact,
the theory is if you don't eat it, it can't be a problem, but you don't miss the obvious. We may be missing an opportunity. All right. Oh, we didn't quite answer the question.
All right, now I know what to tell patients.
Yeah, go see Frank.
And I still know where I stand.
Go see Frank.